Sathyabama Institute of Science and Technology.
Good morning, everybody. This is Jesia Persis Preethi, Assistant Professor from Department of Biotechnology, Sathyabama Institute of Science and Technology, deemed to be university. Today, we'll be discussing about gene and central dogma and gene expression studies. What is a gene and why should we study about gene and genetic engineering, and what is the science behind gene and understanding the gene? So, for all the questions, maybe we'll be finding the answers in the current class. Gene is the basic physical and functional unit of a hereditary, and Gregor John Mendel has contributed a lot for the science of genetic engineering, and therefore he is known as the father of genetics. And he was the one who came up with the laws of inheritance and has done an extensive study about understanding genes. And in this particular aspect, in this particular class, we'll be learning more about gene and the molecular level of understanding what is the reason how the inheritance studies make a very great impact on the today's life. In 1909, a Danish botanist Wilhelm Johnson coined the term gene, and he said that the physical and functional unit of hereditary is nothing but gene. And if you see, gene is nothing but a part of DNA, and gene specifically codes for protein, and we have a lot of genes present in our human body, and the genes generally, the number of base pairs which make a gene can be sometimes a few hundreds to sometimes even 2 millions of base pairs form a single gene. And not entire DNA makes an impact, and not an entire DNA results in the formation of gene. Rather, part of the a very little part of the genome only acts as a gene and helps in inheritance. And alleles are the alternative forms of gene. And here alleles are nothing but we inherit traits from our parents, the physical and genetic traits from our parents. So alternative forms of genes, they are inherited from our parents, and when there is this haploid set of chromosomes which are inherited, when they undergo crossing over, they result in the formation of diploid genome. So uh, if you have to thank and if you have to appreciate the efforts, it is Francis Crick. We know about Watson and Crick, who together came up with the double-stranded helical structure of the DNA. So Francis Crick is the one who has termed or named this particular science behind understanding how gene results in the formation of protein. For all this, Francis Crick and his contributions are very, very crucial. The central dogma of molecular biology was put forth by Mr. Francis Crick, and in the year 1958 he proposed this phenomenon called as the central dogma of molecular biology. And Francis Crick, in his own words, he says, I called this idea the central dogma for two reasons. I suspect I had already used the obvious word hypothesis in the sequence hypothesis, and in addition, I wanted to suggest that this new assumption was more central and more powerful. So, quoting his own words, so he was the reason, he was the one who named the entire concept as central dogma, meaning that how a gene results in ending up becoming a protein which is expressed as a physical trait. The concept, the basic idea behind the central dogma is how DNA makes RNA and RNA in turn makes protein. So, though all the DNA codes, the genes code for protein, we can see that DNA do not directly code for the protein, rather DNA is resulted in the formation of RNA, specifically messenger RNA, and the code which has the messenger RNA, which is written in the messenger RNA, is transferred to the protein. DNA makes RNA and RNA makes protein. This is the central idea, and this is how all the living organisms survive. And if you see, DNA doesn't directly code for protein, rather there is a intermediary molecule which is RNA. And RNA, the need for RNA, if we have to emphasize, say, in order to protect the DNA or the genome from enzymatic reactions which are present in the cytoplasm, a DNA is well encased within a nuclear membrane. And in the nucleus, it is well protected so that there is not any enzymatic reaction which will cause any deformation in the DNA. Therefore, it is present in the nuclear membrane. And now, this DNA is a double-stranded heavy molecule which cannot move through the nuclear pore. Therefore, we have this DNA which is double stranded, which is present in the nucleus, and we know that proteins are coded in the cytoplasm where ribosomes synthesize or biosynthesize your protein. Now, DNA cannot move from the nucleus to the cytoplasm, and neither the protein making complex cannot enter the nuclear membrane. Therefore, we need an intermediary here, which is RNA. Now, DNA, once it transcribed into an RNA, which is generally called pre-RNA, this pre-RNA moves from it's a single-stranded molecule, therefore it can very easily move to the cytoplasm through the nuclear pore. And once it moves, it undergoes lots of post-transcription modifications which will result in the formation of mRNA. mRNA codes for the protein. Now the protein is synthesized based on the code which is written on the mRNA, and with the help of tRNA and ribosomal complexes, protein is formed. So this is the basic idea about the central dogma, and we can see in the current picture, DNA is replicated to form copies of DNA, which is a semi-conservative methodology. And this DNA is transcribed into an RNA, and the process is called transcription. And from the RNA, it is coding to the formation of protein, which is called translation. And for protein formation, there is requirement of mRNA, a ribosomal complex, and a tRNA. All put together, this is how the central dogma plays a predominant role. And the next concept that we will be seeing is gene expression. Gene expression, how does a gene express itself as a physical trait or a physical characteristic? It could be either an enzyme or a cell surface integrated molecule or integrated protein or any other physical trait for that case. So first and foremost, if we can see, DNA generally transcribed into forming different types of RNA. It could be a ribosomal RNA, or it could be a tRNA, which is a transfer RNA, or it could be a microRNA, which is miRNA, or it could also be a small nucleotide RNA, snRNA, which it's also known as small nuclear RNA. And the other RNA which is formed is generally the messenger RNA, which can code for a protein. And rRNA, tRNA, mRNA plays a very crucial role in forming the translation complex, which will help in translation of the protein, that is in the biosynthesis of protein. If you see this miRNA, snRNA, that is microRNA, and small nuclear RNA, they are very crucial for protecting the mRNA. Say snRNA plays a very important role in formation of the spliceosome and the post-transcriptional modifications of pre-RNA into mRNA. And similarly, miRNA, which is a microRNA, helps in protecting the RNA from denaturation as well as unnecessary translation of the protein. Means we need the protein to be expressed only at one point or the other. We do not need it all throughout the cell cycle. So whenever we don't need it, miRNA binds to the mRNA, which will help in preventing the translation. So the expression of eukaryotic gene is a very complex process which will have all these various stages forming, and finally a protein is formed, and the protein generally ends up in metabolic enzymes, cell structure, or even signal transduction. So this is how a gene expression takes place. Gene expression refers to the events that occur during the transfer of the information from the gene to the protein. And all the process, all the stages which are involved in this process are called as gene expression process. And although the functional product is protein, as we have already discussed, RNA is an intermediary molecule which is very crucial in the translation of protein. And the reason stating that DNA actually is transcribed into mRNA. Now, there are triplet codons which are present which will code for the protein to form, and the thymine which is present in the DNA is translated. When it's translated, it is replaced by uracil, which is another nucleotide, and this uracil A, U, G and C, they form triplet codons which will result in the formation of proteins, which we'll be discussing in detail. So as we were already discussing, so the DNA, and when it is transcribed, it forms a pre-RNA, and the pre-RNA, after it is converted, after it is post-transcribed, it will be moving from the nucleus into the cytoplasm, and in the cytoplasm is where the translation process occurs, and this involves the crucial two phases, which is transcription and translation. So transcription, translation put together ends up in the formation of the mature protein, or this continuous process, the entire process where a gene is being transcribed into RNA, that is a DNA being transcribed into RNA and RNA being translated into protein, the entire phase we call it as gene expression. And we need a lot of enzymes like polymerases, DNA, RNA polymerases, as well as ribosomal complexes, which play a very, very predominant role in the formation of a mature protein. Gene expression involves these phases as we were discussing. The first and foremost, transcription, where the DNA is transcribed into a RNA product which is called pre-RNA, and the pre-RNA, it undergoes post transcription modifications where it is converted to a messenger RNA, where it undergoes RNA processings like removal of introns and joining together of the exons together and splicing of the three terminus, and which results in the formation of a mature RNA. And mRNA moves from the nucleus into the cytoplasm and it is translated into protein. So these are the phases which are involved in the gene expression. And this is a picture which just gives a brief overview of prokaryotic and eukaryotic process. Since prokaryotes do not have nucleus, the entire process, transcription and translation happens simultaneously. And if you see, the genome is directly being transcribed as well as means resulting in the formation of mRNA, and the mRNA simultaneously is translated to form proteins. So since the genome is present in the cytoplasm, the process happens simultaneously. Whereas a eukaryotic genome has the process where it happens step by step, where the DNA is transcribed and the mRNA, after it is undergoing post transcription, it ends up in the cytoplasm where the protein is synthesized. And here are some helpful resources, and I'll be sharing them in the description. Thank you.